Welcome to the Once Changing the World, India's first Future Tech Meets Sustainability podcast. I'm your host, Eddie Havel, and today I'm delighted to have with me Mr. Viraj Mehta, who's a scientist at Psi Life Sciences, where he leads 3D liver spheroids and liver on chip study. He specializes in the area of 3D bioprinting, 3D spheroids, and tumor on chips for personalized medicine and cancer. So, Viraj, really appreciate you taking time and being part of the Humble podcast. Would be great if you could start with the background and your works.
Yeah. Thanks, Eddie, for inviting me for this podcast. And first of all, I would like to congratulate you for coming up with so many amazing topics and so many thought-provoking topics, actually, and inviting so many amazing speakers. My PhD was focused on microfluidics. It's a broader area of microfluidics where I worked on developing tumor on chips devices for personalized drug screening based on patient-derived samples. Before pursuing my PhD, I completed my bachelor's in mechanical engineering. So during my bachelor's in mechanical engineering, I got interested in biological field, and I found that there are so many applications of mechanical engineering, mechanical engineers that uh, those application actually attracted me towards this biofield. So right now I am associated with a CRO called SciLife Sciences, which is a very well-known CRO in India as well as globally. Here we are first time starting advanced 3D culture models that are becoming more and more relevant in the drug discovery field. That are, as you mentioned, 3D spheroids, 3D organoids, and liver on chips. So this kind of models are the future models of drug discovery. They are going to disrupt the drug discovery. How we are doing right now, it this kind of models will change the drug discovery process significantly in the future.
The whole way where we used to, the pharma industry would do the drug discovery process is completely being upended. I think it'll be great if you could, you know, set the ground as in to with the audience, and explain to them what the concept of liver on chips is and what is the purpose that it serves.
You know the drug discovery process. It starts with we start with around 10,000 to 1 lakh compounds, and gradually as we come towards clinical phase, we are left with very few compounds. So when we do the in vitro drug discovery process, usually all the assays that we do, for example, the drug, when you, for example, if you take any oral drug, then it gets absorbed in the gut. So to check the permeability of the drug molecule in the gut, we use 2D monolayer based models. So basically, this 2D models are conventional culture models that has been used widely, and they are reproducible, they are repeatable, but at the same time they lack the the human microphysiological relevance. Because if you talk about liver, so the liver hepatocytes, the main parenchymal cell of the liver, they are kind of in a 3D microenvironment. So they are stacked upon each other in the liver. So how can we mimic this three dimensionality in our in vitro science? So to do that, people have come up with this kind of advanced 3D models like organoid spheroids. And so I would say, like, if you just draw a graph with x-axis as a complexity of the model, then spheroids will be at the left side, organoids will be somewhere in the middle, and liver on chips at the rightmost side. So it constitutes the highest complexity that you can imagine in vitro science. So here you have a 3D culture of the liver hepatocytes inside microchannels. So as the name suggests, microfluidics. So the channel dimension are at a micron levels. So what we do is there are channels inside a chip, a kind of a handheld chip, which can be of this size, which can be made up of various polymers, and using various specialized manufacturing methods, we make this kind of devices in which there are channels in which we inject the cells, and cells are cultured in the inside those microchannels under perfusion dynamic flow. So This is the second component. So first was 3D culture, how to mimic the three-dimensionality. Now I'm coming to the second aspect of the same of the mimicking the liver microorganization, that is the dynamic perfusion. So to mimic the systemic flow that is going through the microvasculature of the liver. So we are also incorporating this dynamic fluidic flow. So this dynamic fluidic flow basically applies various mechanical forces, and this leads to activation of various mechanotransduction pathways. So when you grow this liver hepatocyte outside your body, so it's very challenging to culture them for a longer time with high viability. So you need to make sure that you providing all the growth factors that are natively provided in the liver through the microvasculature, as well as you provide this dynamic shear forces to the liver hepatocytes, and at the same time you also give them a three-dimensional environment to grow. So I would say these three aspects are what differentiate liver on chips from the other 2D monolayer based conventional models. So that is about liver on chips.
Viraj, will be also great if you could maybe talk to us about the significance of it. When you kind of create these liver on chips, what's the purpose that serves in the drug discovery field? What's been built out of the learnings of from these liver on chips?
Yeah, so that's a really good question. So in a one word answer, I would say better drugs. We are making better drugs, because traditionally drug discovery has been it's first of all, it's a very expensive affair, and drugs have been traditionally failed after moving through the clinical trials. So when you fail in the clinical trials, you cannot imagine how costly that is for a drug discovery company. So the idea is to fail fast and fail early. So if you fail early in the preclinical discovery, it's still okay. But when you fail in the clinical trials, so for example, you have come up with a drug molecule and it's showing it didn't show any toxicity when you are doing preclinical animal testing, but when it shows toxicity in the patient trials, then you have to drop that molecule. So that's a money loss basically for the company. So the whole idea of coming up with this kind of models, 3D models, is to fail fast, first of all, fail fast, fail early. Second thing is to predict the human relevant aspects of the drug molecule. So there are many publications that have come now in the you can see many publishing houses have published this kind of studies. So these molecules, they are more predictive of the human toxicities compared to the animal models and other 2D conventional models. In fact, there are many reports that have been published that also we can search. So first thing is, if the drug molecule that is showing toxicity with this kind of models, then you can make an informed decision that you shouldn't go forward with this molecule to patient clinical trials. And if it is not showing toxicity, then of course you can trust this human relevant system and you can go forward with the patient clinical trials. So basically, these models use the human relevant cells. So conventionally, again, animals are the gold standard right now, but animals are not humans, right? Animals, there are, for example, in the liver, there are many SIPs that differ from our liver to the animal. So SIP are the liver metabolizing enzyme. So this enzyme differs from animals, and our enzymes are totally different. So the idea is to use the human derived hepatocytes in this kind of relevant 3D models, so to make the informed decision and to take the better candidates forward towards the clinical trials. So that is the whole idea why people have started using this kind of models instead of animal models.
Right, right. Can you share some of your works related to 3D liver spheroids, and maybe also talk about organoids, because that's a topic I'm like really fascinated with, that how we can now take a cell turn it into a induced pluripotent stem cell, and on a petri dish you can culture that cell into forming whatever you want. I mean, like for example, if you take in like a stem cell of a brain, you can actually create a brain organoid, or you could create a kidney, you could create a liver, and so on and so forth. Maybe talk about your works in the space of 3D liver spheroids and also organoids. And do you see that in the future that maybe we could someday possibly grow, a full blown organ? Would that be a possibility?
So, first of all, let us define what is spheroids, what is organoids. So, spheroids are kind of a forced assembly of the cells, I would say. So, we grow this kind of. We first of all have a single cell suspension that we grow in ultra low attachment plates. So that is called ULA plates. So that plates basically don't allow cell to adhere on the surface, and instead of adhering to the surface, they form 3D assembly. So that is a 3D spheroid. So it's a spherical shape. So that's called 3D spheroids. On the other hand, organoids are more advanced models, as you said, using induced pluripotent stem cell and not even induced pluripotent stem cell. You can also use the organ resident stem cell as well, and organ specific parenchyma as well to make the organoids. So when you grow this iPSC derived cells inside a matrix called matrigen or other synthetic matrix, naturally derived matrix also you can use. So when you grow them inside this matrix, they divide and they form kind of a 3D, I would say, aggregate or an assembly of cells. So that is called an organoid. Now, organoids are also, they have a capability to recapitulate the organ-specific structural characteristics, organ-specific cellular population, all the organ parenchyma and stromal population also they can incorporate. Further, you have also capability to grow vasculature inside organoids. So now imagine how beautiful that is. You have a all the organ relevant cells that have differentiated in the organoids, and you have also vasculature in the organoids. So this is like a a mini, I would say, if you are considering liver organoid, then it's a mini liver tissues that you are growing in your lab. So that's the beauty of organoids. Now, coming to your question, whether a full thickness organ will be possible or not. So 3D bioprinting is one of the techniques through which this kind of organ transplantation and organ printing, uh, you know, uh, uh, future is possible, because the 3D bioprinting has a capability to deposit cells in a well-defined pattern. So I would say 3D using 3D bioprinting and these organoids, we can make kind of a mini organ, but full thickness organ is, I think we are still far away from that. However, I have seen some of the startups that are working in bone tissue, engineering. So they have come up with, so they using microcity data, they get the idea of the defect site, and they print the bone tissue, exactly matching that size, and you can transplant that to the patient. So for bone, I have seen a few startups that are working in the space, but when you talk about liver, heart, these are some of the liver, heart, kidney. These three organs are actually, there is a scarcity of the donors also. So of course, research is going on, but I mean, we are still far away from that. Growing a full thickness organ, we are definitely far away from that.
Can you share what's the cutting edge of innovation in the field of 3D bioprinting currently? And maybe some of the works that you've done in the 3D bioprinting space. What can we currently 3D bioprint, from skin, organs? What can we do at this point in time?
3D bioprinting, first of all, I would say the load-bearing tissue, the bone, cartilage, muscles. These three organs, people have come up with 3D bioprinting techniques, how to print them in a petri dish. Uh, s- uh, s- and in my PhD time, some of my colleagues were working on 3D bioprinted blood vessels. So what we do is we from the patient-derived imaging data, we can feed that data to a 3D bioprinter, and it basically converts that data into G codes. And the printer will basically follow that G code, and it will print the specific that same shape of the blood vessel. So that blood vessel, when you 3D print, you have to be very aware that that blood vessel is still not matured enough. So to mature that blood vessel, we used to, use a kind of a bioreactor. So that bioreactor basically applies mechanical forces to this 3D bioprinted blood vessel. So mechanical forces in the sense it will actually dynamically perfuse media, cell culture media through that blood vessel, and with time the blood vessel relevant markers and its mechanical properties will enhance. So basically, we are maturing that tissue before transplantation. Of course, we haven't reached the transplantation stage, but we have done till this stage printing of the blood vessel using patient-derived cells and maturing it in the bioreactor. So the point that I want to make here is the bioreactor forces are very important. Although we think that we will print the tissue using bioprinting, but we need to mature that tissue before. And for load bearing tissue, that is more relevant. So for example, if you are thinking of printing a tendon or muscle or cartilage, then after printing you have to apply physiological forces, and you have to mature that tissue so that its functional characteristics, its mechanical properties come very closer to the human cartilage tissue, and then only you can go for the transplantation in the patient.
What are the things that can be done at this point in time? And what do you think we could be able to print maybe in the next few years?
Yeah, so as you told, the bone is a very widely, I would say, used application of 3D bioprinting, because you have a CT data available very nicely from the patient defect side. So people have come up with the facial surgery when because of the accident, if you lose some of the part of your face, then using the CT data, you can print that particular defect site using bioinks. So bioinks are biocompatible materials. So those materials, using those materials, you print the defect site, and you can fix that particular defect site in the patient. So bone is I would say we are at an advanced stage for the bone. Uh, and for heart, I think heart 3D bioprinting of the heart is very challenging, but some of the researchers in US and Europe are doing it. In fact, there is already one publication, I think it came 5 to 6 years ago, where they actually demonstrated beating heart, 3D bioprinted beating heart. So those kind of things are possible, but the size of those printed tissues are very small. So as you told, the full thickness, they are not ready to be transplanted for a patient-derived application. So, we are still very far from growing, full thickness organs. And I have also seen some of the publications that have come up in the lung bioprinting area. So people have started, researching in this area as well. Uh, and apart from that, there are kidney 3D bioprinted models also, but they are not meant for tissue regeneration, tissue engineering application. They are meant for drug discovery application. So 3D bioprinting is used widely for tissue regeneration, tissue engineering, but it is a mini use for the drug discovery application. So bioprinting has a immense applications in terms of tissue regeneration and drug discovery applications.
You have also been doing some works on tumor on chips. Can
Yes,
you talk
yes.
about a tumor on chips and how is it being leveraged in building personalized therapy?
Yeah, so what we were doing is we were asking ourselves whether because cancer is first of all highly heterogeneous in nature. So one size fits all approach doesn't work anymore in cancer therapeutics. So every patient, histopathologically same cancer. If the patient set of patients that have been diagnosed with the histopathologically same cancer, but still, you know, if you administer the same, drug regimen to all the patients, the responses will be different. So how to come up with solutions which which can come up with suitable drug therapies for each and every patient. So that's the whole idea. So using this microfluidic approach, so we actually designed a microfluidic device, which for which we also obtained a patent. So in which we were looking to screen three different anti-cancer drugs and the respective combinations. So in cancer, another thing is instead of going for monotherapy, the combinatorial therapy is much better, because in combinatorial therapy means you are administering multiple drugs together, so to have a higher efficacy and to lower the drug concentration, because you know that cancer drugs have also off target toxicities. So the hair loss and everything. So with conventional chemotherapy, the fastly proliferating cells also die, in addition to the cancer cells. So we were looking for selecting combinatorial drug therapy for each and every patient. So we had three patient-derived oral tumor samples, oral cancer samples, and we isolated oral cancer cells from those three patient-derived tissues, and we grow them inside microfluidic devices in a 3D spheroid format. So in microfluidic devices, you can grow the tissue in 3D spheroid, you can grow in 3D organoids. So there are a number of possibilities are there. So basically, the organ on chip devices, they are compatible with wide variety of 3D tissue model as well as 2D culture methods. So in our case, we design our microfluidic device such a way that it can form our cancer cells can form 3D spheroids inside microfluidic device. So after we seed those patient-derived cells in our device, they will form spheroids within 24 hours, within 1 day. And after 72 hours, we dynamically perfuse the three drugs. So these three drugs, we selected those three drugs based on the well-known oral cancer chemotherapy regimen that is available online, that is approved therapies that are available online. So those therapies, when we applied for all the three patient-derived tumor samples, we found that the drug responses were highly heterogeneous. So first thing, we could identify which drug combination is working the best for the patient one, for the patient two, and patient three, and which drug combination will not work for patient one, patient two, patient three. So every patient, they all three were diagnosed with oral cancer, but all the three shown different drug responses on a microfluidic device. So we envision that using these microfluidic devices, one day we will be able to identify the perfect therapy for each and every patient, each and every cancer patient. So that was the whole idea of my PhD work. Yeah.
Right. Will be great if you could share, what are the challenges that you have faced in designing or fabricating these organ on chips, and how do you overcome these challenges to ensure that these stay robust and market ready?
Yeah, so first thing is this organ on chips devices, their throughput is very low. Throughput means, uh, if you go with the conventional 2D monolayer based models, for example, you take a 96 well plate. So in 96 well plate, you can screen number of drugs at a time. On the other hand, the microfluidic device, you can screen only three drugs at a time on one patient derived tissue. So first thing is how to improve the throughput of this kind of models. That is one big question. Because again, when you talk about drug discovery companies, they are looking for high throughput. They are looking for high throughput as well as complexity. So they are looking for a balance between these two aspects. So when you come up with a microfluidic device which is very low throughput in nature, of course, it won't attract them. So first thing is we need to come up with a design which can make this models high throughput in nature, so that we can screen maybe 20-30 drugs at a time on one patient derived tissue. Second thing is the microfluidic devices are traditionally manufactured with silicon material called PDMS, polydimethyl siloxane. So this material, was introduced in 90s by some of the researchers, and we have been following that material to this time. So that material has number of advantages. That's why people are still using it. That because it's first of all it's a very transparent material. So because of transparency, you can do a very nice microscopy of the cells. The cells will be nicely visible. You can track the cell development with time. Second thing is they are PDMS is biocompatible in nature. It doesn't show any cytotoxicity to the cells. So that is the first criteria. When you are using any material with the cells, the material should be biocompatible. And third, a very important property of PDMS is it's gas permeable. So it allows the exchange of oxygen and carbon dioxide. So that is another added advantage for the cell, for the longer cell culture in this kind of systems. Uh, but at the same time, when you talk about drug discovery related application, the PDMS is known to absorb this kind of drugs on its surface. So people have come up with some alternate choices of the material, some alternate fabrication methods. So conventionally, this PDMS microfluidic devices has been are manufactured with a method called photolithography. So which again very time consuming process, and it requires a lot of steps also. But on the other hand, people have come up with alternate fabrication method. One of the fabrication method is 3D printing. So using 3D printing, people have shown that you can make your microfluidic devices in 1 or 2 steps. So, from reducing 10 to 12 steps that are involved in photolithography. This PDMS based microfluidic devices also can be manufactured with the 3D printing. So to reduce the manufacturing time. Now coming to the other materials that are available, alternate for PDMS, because PDMS is known to absorb these drug molecules. So what happens is if for example, if you flow 1 micromolar of let's say some so and so drug, then if some of drug molecules are getting absorbed, then of course your concentration is going to change. So your cells are not going to be exposed with 1 micromolar concentration. It will be exposed with some lesser concentration. So your results will also get affected. So people have come up with other polymers like polycarbonate, polystyrene, but this polymers can be manufactured with a very high-end manufacturing method, which may not be available in academic settings. So these materials are there, for us to explore further. But the usual, the usual process of developing microfluidic is you start first with PDMS, you show the proof of concept on PDMS, and after that, once you are confident that my microfluidic device is working, then you go forward with this industrial material like polystyrene, polycarbonate, and you go with the industrial manufacturing methods. So those methods are again very high throughput in nature. So you have a very high, manufacturing time is very low. So this would be an ideal process of from designing a microfluidic devices towards commercializing your microfluidic device.
What do you think the future holds for this organ on chip technology? And are there any emerging trends or advancements that have caught your attention in the field of organs on chip technology?
We are not far from that stage when we will come to know whether industries are all in or they are all out, whether they are going to use this organ on chips or they are not going to use this organ. So we are not far from that time point. We'll soon come to know whether, because there have been so many publications that have come up so far, targeting each and every combination that you can think of. Not only single organs. People have come up with multi organs on chips as well. First thing is, the multi organ on chips is very important to develop, because when you are thinking of using this organ on chips, you are thinking of reducing or replacing the animal usage. So how to mimic the overall body system in an in vitro system. So multi-organ on chips is one of the I would say a future trend that I can see. People will start going towards establishing multi-organ on chip devices. Single organ on chip devices, they have been established very nicely. Second thing is how to test the newer drug modalities that are coming up in the market. So far we have mostly tested small molecules drugs using in vitro models. But there are newer drug modalities that are coming up like proteins, biologics, antibody drug conjugates, peptides. So this all these newer drug modalities come up with their own challenges. So how to overcome those challenges using this kind of advanced 3D models will be very interesting to see, and I'm looking forward to it how people will use this kind of model to test this kind of modalities. Third thing is, as I told you, the drugs are still failing because of the toxicities. The toxicity is a very big challenge for any drug discovery companies. So that's why apart from predicting the drug clearance, we are also looking at whether can we predict the liver toxicities as well using this liver on chips models. So that can inform our clients that yes, this candidate can be potentially toxic in the human. So establishing drug-induced toxicity models is also one of the future trend that I can see in this space. And you know Fourth thing is, people have come up with disease models on chips as well. So recently, I was reading one paper where they have recapitulated the compromised blood retinal barrier in case of diabetic retinopathy. So in diabetes, people also can undergo vision loss. So people have come up with these disease models to using transcriptomic data. The drug discovery companies are identifying newer drug targets. So how beautiful it can be. So when you can mimic the whole disease pathophysiology on a organ on chip, now you have that disease in your hand, and you can using transcriptomic data, you can come to know which gene is getting overexpressed, or which gene is getting underexpressed. So based on that, you can identify newer drug targets, and you can start developing drugs that target that kind of drug target. So these are the four areas I see where this organ on chip field will move on.
Viraj, really appreciate you taking time and being part of the podcast. My last question to you: what are the timelines you see this being democratized? Where everyone can have access. Anybody who's got a specific healthcare issue has access to this. This whole personalized medicine that you're talking about, a chip that you create which kind of simulates your organs, and you learn from it, and you do drug discovery and stuff like that. When does this become like market ready? And also maybe kind of paint a picture of what the world you want to see in the next 5 to 10 years. How do you see healthcare evolving in the next 5 to 10 years with these kind of technologies, organ on chips, available for everyone?
The one of the biggest barrier right now is the regulatory aspect. We need to see, regulatory aspect, because, for example, even if you use this kind of models in your drug discovery application, how the, for example, if it is developed in US, then how FDA is going to, review that data. So there are, lack of guidelines, as to how to use this model, in what scenario you need to use this model, and what kind of what performance parameters that you need to set before using this kind of models for drug discovery application. So uh, the organ on chip makers, as well as the researchers and the regulatory bodies, has to come up and work together to solve this kind of issues before widely using this, or maybe regularly using them in the drug discovery application. That's the biggest barrier I see. Otherwise, the minor issue that I feel is the repeatability and reproducibility of the data from this kind of models. Because again, in drug discovery, if in industry, if your model is not robust enough, if the data is not repeatable, reproducible, then you cannot make any informed decision from that. So, all the commercial organ chip makers has to come up with good publication showing, that your data is robust enough, your data is repeatable, reproducible. So to enhance confidence in this kind of this all these big pharma companies. So right now, most of the pharma companies are still in a confusion state whether to use them or not to use them. They are still waiting for the right time, I would say. Although there are publication, but they are all again, they are all, I would say, very they are not they are kind of exploratory studies that they have done. It's not like they have come up with a drug candidate and that got that they filed for new drug application using this kind of models. So that this is what I feel that we are still far away from that. Coming to your second question, how I would like to see the future. I when I finished my PhD, I made this goal of my career that I want to make the best model that can be used for drug discovery, so to reduce and replace animal usage in the drug discovery process. So I'm working towards this noble cause, how to replace and reuse or uh, kind of you know reduce the animal usage in the drug discovery. So I would like to see the future where this kind of models are being used regularly in the drug discovery process, and it can lead to reduction in animal usage as well.
Viraj, really, really appreciate you taking time being part of the podcast. More power to you. Yes, I think these are still in this very nascent stage, early days. But yes, I guess, when this kind of matures, there are a lot of animals that which get mistreated, and these experiments that's being done, which obviously has helped the human race produce these better drugs and better healthcare solutions. But it's about time that we as humans find other areas and other ways to kind of discover drugs without harming these animals. And I think
Correct.
we sitting in such a fantastic point of time. I think all these tech stack is converging, and these through these tech stack we will get into an area where we will possibly get into personalized healthcare, and through these organ on
Yeah.
chips technologies and AI being leveraged for drug discovery, and biotech with genomic sequencing and things like that. I think we sitting in a fantastic point of time. So more power to you, and wish you the very best. I hope the regulatory hurdles minimizes, and these technology stacks becomes more widely used in industries, so that those these things which are now being experimented on in the labs and in these pharma companies reaches the normal people who are waiting for solutions, so that their health can be better. So more power to you, and I really appreciate you taking time and being part of the podcast. To my listeners, if you like what you see in here, then please press the subscribe button, and until next time, see you guys. Bye bye. Thank you, Viraj. Really appreciate this.
Thank you, Eddie. Thank you so much. Thank you.